Welcome back to our introduction to bioinformatics. Previously, we mentioned how bioinformatics is a tool used to interpret vast amounts of biological data. In this video, we will be diving into areas within genetic testing, exploring its significance and how bioinformatics helps decipher genetic code. Join us as we also discuss patient and donor data pathways and how we protect this sensitive data. Imagine DNA as a recipe book that's passed down in your family. Just like a recipe book contains instructions to make different dishes, DNA contains instructions to make you who you are. It's written in a special code, not with letters and numbers, but with 4 chemicals that scientists abbreviate as A, T, C, and G. These chemicals arrange in a specific order, just like you would have different steps within a recipe. These steps tell your body how to build and maintain itself. This DNA recipe book is really long, so to break it down, it is split into chapters, which we call genes. Each gene has instructions for one small part of you, such as the colour of your eyes, the type of hair you have, or even how your body fights off illness. Everyone's DNA is mostly the same, but the small differences make each of us unique. Sometimes, there can be a typo or error within a recipe. In DNA terms, these typos are called mutations. Some of these mutations don't change much. An example would be if you accidentally added an extra spoon of sugar to a cake recipe. However, others can be more significant and might lead to health problems. Doctors and scientists use DNA to understand more about your health. For example, to identify if you have a higher chance of certain diseases, or which medicines might work best for you. That's why DNA is so important. It's the book of you. When scientists do genetic testing, they take a small sample from your body. Common examples are blood or saliva. They then look at the sample to read parts of your DNA. This is like checking certain pages of your book to understand more about your health. We can also diagnose viral and bacterial infections using genetic testing. Remember, what is found within your DNA is just one piece of the puzzle. Your environment, lifestyle, and many other factors can also affect your health. Genetic testing can provide us with important information, but it's not the whole story of your health. Blood cancer, or blood disorder, is linked to changes in your body's cells. These changes are often connected to your DNA, which is your body's blueprint. Genetic testing is all about reading this blueprint to get specific information about your health. The main reasons why genetic testing is important include understanding your disease, choosing the right treatment, predicting how a disease might progress, targeted therapies, and family information. An example of a condition requiring genetic testing is sickle cell disorder. This is an inherited condition which affects red blood cells. The red blood cell becomes sickle in shape, which leads to early destruction and blocking of blood vessels. This condition is more often seen in people with African or Caribbean family background. Due to early destruction of red cells, patients often need blood transfusions to replace the sickled cells with normal cells. First, a blood sample will be sent to NHSBT for extended blood typing so that we can establish the blood group of the patient. Some patients have rare variant blood groups which require further analysis by specialist genetic staff. This can include additional testing to read the genetic code of the red blood cell to determine what type of blood is most suitable for the patient before the next transfusion event. Information about the patient's blood group is sent to the National Register for Sickle Cell Patients. This ensures specific blood requirements for these patients can be accessed from any hospital across the UK. In the context of stem cell therapy, bioinformatics contributes significantly to the typing and matching processes between donor and recipients, ensuring the compatibility of stem cells. Stem cell transplantation is a medical procedure used to replace damaged or diseased cells with healthy ones. At the heart of this process is the need to ensure that the donor's cells are compatible with the patient's immune system. This is where HLA typing and HLA matching comes in. HLA, or human leukocyte antigens, are structures found on the surface of cells that are key to our immune system's recognition process. HLA typing is a critical laboratory test to determine compatibility between a donor and a recipient for organ transplant or stem cell transplantation. Bioinformatics helps in analysing and comparing these HLA proteins between the donor and recipient. Through complex data analysis, bioinformatics can accurately identify the HLA types, which are incredibly diverse and vary greatly among different individuals. This matching is essential as a close HLA match between donor and recipient greatly increases the chances of a successful stem cell transplant. By utilising advanced computational tools and algorithms, bioinformatics streamlines the process of HLA typing and matching, making stem cell transplants safer and more effective.
In the world of hematopoietic stem cell transplantation, bioinformatics is an indispensable tool for ensuring successful outcomes. Engraftment marks the moment when transplanted stem cells take root and begin producing healthy blood cells in the patient's body. Bioinformatics plays a pivotal role by analysing genetic data to assess how well donor cells will integrate into the recipient's body. By examining specific genetic markers, bioinformatics predicts the likelihood of engraftment success, a crucial milestone in the patient's recovery. Furthermore, bioinformatics contributes significantly to understanding and enhancing survival rates post-transplant. By analysing vast clinical data, it identifies patterns and factors influencing long-term survival, such as patient response, risk of complications, and post-transplant care effectiveness. This invaluable information empowers doctors to tailor treatment plans, maximising the chances of a successful outcome for each patient. In essence, bioinformatics is not just a tool, it's a lifeline for patients undergoing stem cell transplantation, improving engraftment outcomes and overall survival rates. With bioinformatics, we're shaping a future where every patient has the best chance at a successful recovery.
So how do we store and protect patient and donor data? NHSBT is required to keep clinical data for 30 years after collection, which is necessary for donation tracing and clinical patient management. If a patient or donor consents, this data can also be used for research so that we can help to improve our services. Nevertheless, some people may find long-term retention of the data concerning. NHSBT adheres to the Data Protection Act 2018 and GDPR requirements, as well as the Freedom of Information Act 2000. This means that data will be used only for the designated purposes, and donors and patients may request data held by NHSBT. To keep patient and donor data safe during transfer and storage, it is encrypted and can be anonymized. Encryption is a means of making data unreadable by distorting it. This means the data goes from looking like this to this. This stops others from being able to read patient or donor information. When staff need to access the data, it is decrypted again using a key. Patients and donors are also given ID numbers. This means that most routine use of their data is anonymized, with patient and donor names only being accessed by certain trained staff in circumstances where the individual is to be contacted. In summary, your DNA can be likened to a unique recipe book, which guides who you are using the chemicals A, T, C, and G. Your genes are chapters within the book detailing your personal traits. Genetic testing, akin to reading the recipe book pages, helps doctors to understand your health, predict diseases, and tailor treatments. In blood cancers, it's vital, offering insights for targeted therapies and family health history. We hope you found this information informative. Thank you for watching.